Welcome back to O-Level Oasis, your go-to guide for tackling CAIE exams with ease. Today, we're continuing our journey through O-Level Biology 5090, and just so you know, all the topical notes for this chapter are available in the video description and in the pinned comments for your convenience. Right now, we're focusing on Chapter 8, Human Nutrition. So far, we've completed 2 units. First, we studied diet, and then we explored the human digestive system. Now, it's time to move on to the third and final unit of this chapter. This unit is Unit 8.3, Absorption and Assimilation. Here, we'll be looking at what happens after digestion, the processes that take place, the organs involved, and exactly how absorption and assimilation occur in the body. We'll follow the syllabus closely as we go through this topic, step by step, so let's dive in and get started. First, we have specification number 1, which asks us to state that the small intestine is the region where nutrients are absorbed. This simply tells us that nutrient absorption occurs in the small intestine, and we will explore this in more detail to understand it better. So, let's begin with the definition of nutrient absorption. The key definition is that nutrient absorption is simply the movement of digested food molecules from the digestive system into blood and lymph. In simple terms, this is the process by which the nutrients from the food we eat pass into our bloodstream and lymph so our body can use them. Nutrient absorption involves the movement of digested food molecules from the digestive system into 2 main transport systems. Blood. This carries glucose and amino acids. In other words, these molecules enter the blood after digestion. Lymph. This transports fatty acids and glycerol, which are absorbed into the lymphatic system. Location. Nutrient absorption occurs mainly in the small intestine, specifically in the ileum. Methods of absorption. There are 3 main ways nutrients are absorbed: diffusion, osmosis, and active transport. So, the small intestine is the region where nutrients are absorbed. These nutrients then pass through the cells of the intestinal wall into either the blood or the lymph, depending on their type. So, let's take another look at the diagram. Earlier, we identified all the parts of the human digestive system including mouth, teeth, epiglottis, liver, gallbladder, duodenum, ileum, small intestine, appendix, salivary glands, pharynx, esophagus, stomach, bile duct, pancreas, colon, rectum, anus, and the large intestine, including the small intestine. And now our primary focus is on small intestine. If you look closely, you'll see that the small intestine has 2 main parts, the duodenum and the ileum, which connect to the large intestine. This is important because the small intestine is the region where nutrient absorption takes place. So, if we zoom in a little on the small intestine, we can see this diagram that explains how absorption actually takes place. The diagram highlights the structural adaptations of the small intestine that make it so efficient at absorbing nutrients. First, we see the overall structure of the intestine. It has muscle layers and folds inside the lumen, and these folds are formed by finger-like projections called villi. When we magnify further, we can see each villus in detail. Inside every villus, there is a dense network of blood capillaries and a central lymphatic vessel called a lacteal. This arrangement brings the blood and lymph vessels very close to the surface, which makes absorption quick and efficient. The capillary bed absorbs digested carbohydrates, like monosaccharides, and proteins in the form of amino acids. These are then carried through the hepatic portal vein straight to the liver for further processing. Meanwhile, the lacteal takes up digested fats, specifically fatty acids and glycerol, which are transported into the lymphatic system before eventually entering the bloodstream. On the surface of each villus, we also find the epithelium, which is made of epithelial cells. These cells have countless tiny projections called microvilli, forming what we call the brush border. The microvilli not only increase surface area even more, but also help complete the final stages of digestion and absorption. So, overall, this diagram shows how the villi and microvilli of the small intestine massively increase surface area and enable efficient absorption. Nutrients move into either the blood capillaries or the lacteals, and from there, they are transported to the liver or into the lymphatic system. So, let's talk about some specific adaptations of the small intestine, especially the ileum, which is the inner section specialized for nutrient absorption. The ileum has several important features that make absorption both fast and efficient. First, it has a very long length. This increases not only the surface area but also the time available for nutrients to be absorbed as food moves through. Second, the inner lining is highly folded and covered with millions of tiny finger-like projections called villi. And on top of that, each villus is covered with even smaller folds called microvilli. Together, these structures create a massive surface area, making absorption much quicker and more effective. Third, the walls of each villus are extremely thin, just 1 cell thick. This provides a very short distance for diffusion, which speeds up the movement of nutrients into the blood or lymph. Fourth, there is a rich blood supply. Each villus contains a dense network of capillaries that quickly carry glucose and amino acids into the bloodstream. This constant flow of blood also helps maintain a steep concentration gradient, which keeps diffusion happening efficiently. And finally, in the center of each villus is a lacteal, a lymph vessel that absorbs fatty acids and glycerol. These fats are transported into the lymphatic system before eventually joining the bloodstream. So, with its long length, huge surface area, thin walls, rich blood supply, and central lacteals, the ileum is perfectly adapted for rapid and efficient absorption of nutrients.